At the core of human biology lies a fascinating system of chromosomes that determines our sex, specifically the X and Y chromosomes. Each individual typically has 23 pairs of chromosomes, making a total of 46. Among these, one pair is known as the sex chromosomes, which are designated as X and Y. The presence or absence of these chromosomes plays a crucial role in defining whether a person is male or female. In general, females carry two X chromosomes, represented as XX, while males possess one X and one Y chromosome, designated as XY. This chromosomal distinction is established at conception. During fertilization, a sperm cell, which can carry either an X or a Y chromosome, fuses with an egg cell, which always carries an X chromosome. If the sperm contributes an X chromosome, the resulting zygote will be XX, leading to the development of a female. Conversely, if the sperm contributes a Y chromosome, the zygote will be XY, resulting in a male. This simple yet profound mechanism illustrates how the genetic material from both parents combines to shape the sex of the offspring. But how do these chromosomes exert their influence? The Y chromosome is relatively small and contains a gene known as SRY, which stands for Sex-Determining Region Y. This gene triggers a cascade of events that lead to the development of male characteristics. It initiates the formation of testes, which subsequently produce male hormones, or androgens, that guide the development of male physical traits and reproductive structures. In the absence of the Y chromosome, or more specifically the SRY gene, the default developmental pathway is female, leading to the formation of ovaries and the development of female characteristics. To further understand this process, we can delve into genetic diagrams, specifically Punnett squares, which illustrate the possible combinations of gametes during fertilization. Gametes are the reproductive cells, sperm in males and eggs in females, that contain half the genetic material of an individual, or 23 chromosomes. In the context of sex determination, let's consider a simple example where a male with XY chromosomes and a female with XX chromosomes reproduce. When we create a Punnett square for this scenario, we place the possible gametes from each parent along the top and side of the square. The male can produce two types of gametes, one carrying an X chromosome and one carrying a Y chromosome. The female can only produce gametes carrying X chromosomes. This results in 4 potential combinations in the Punnett square: XX, XY, XX, and XY. From this, we can see that there is a 50% chance of having a female, XX, and a 50% chance of having a male, XY. This visual representation not only simplifies the understanding of genetic combinations, but also highlights the randomness inherent in the process of sex determination. The implications of this genetic framework extend beyond mere biological classifications. It raises questions about gender identity, societal roles, and the intricate relationship between biology and experience. While the XX and XY chromosome configuration provides a foundational understanding of sex, it is essential to recognize that gender is a multifaceted construct influenced by a myriad of factors, including culture, environment, and personal identity. In conclusion, the interplay between X and Y chromosomes is a fundamental aspect of human genetics that determines biological sex. The SRY gene on the Y chromosome initiates male development, while the absence of this gene leads to female development. Genetic diagrams, such as Punnett squares, serve as valuable tools in illustrating the potential outcomes of gamete combinations, providing insight into the probabilities of male and female offspring. This intricate dance of chromosomes not only shapes our physical attributes, but also invites deeper contemplation about the nature of identity itself.